And I always loved Wilson, and when he told me back in 2000 that we were losing him from the ALS care community, I was devastated. But it turns out that we're pretty lucky because we have a friend at the FDA who really cares about ALS. And Wilson is a terrific guy. He really cares. When he took care of people living with ALS, you couldn't have found a finer physician or a more caring guy. And he was committed, and he remains committed not only to ALS, but to making sure, and he's going to tell you a little bit about drug development and taking ALS therapies into the clinic today, but he cares and he wants to get those therapies out. And he'll talk to you a little bit about that. He went to University of Chicago, Pritzker School of Medicine. He was co-director when I knew him at the University of Texas Southwestern Clinic. And now he's director of the Division of Clinical Evaluation in Pharmacology and Toxicology at the FDA. And he can probably explain better what he deals with, but mainly cellular tissue and gene therapies and biologics. He's going to talk to us today about drug development and taking ALS therapies into the clinic. Wilson, that's it. Do you need a kickstand?
We'll see.
I'll look to the side in case you don't.
Thank you, Terry.
Thank you.
I really do appreciate the opportunity to speak with you all today. And I'm going to talk a little bit about the FDA and the work that I've been involved with at the FDA. All right, so the FDA, for those of you who are not familiar with what we do, consists of 7 different centers. And that includes, on the far right of this slide, the Center for Tobacco Products. We regulate tobacco products. And we regulate, there in the middle, the Center for Food. We regulate food, and products for animals are regulated in the Center for Veterinary Medicine. But the products for humans, human medical products, are regulated in the 3 centers at the left of the slide. It's the Center for Drugs, the Center for Devices, and the Center for Biologics. I work in the Center for Biologics, so what I'm going to talk about is from the perspective of biologics. Particularly, I work with cell and gene therapies. So that's stem cells, and well-differentiated cells, embryonic cells, all types of cells, and gene therapies. And these are very complex and very exciting products. So I like working there. It's a lot of fun. But we're here to talk a little bit about ALS, Lou Gehrig's disease. And of course, you're all familiar with the story of Lou Gehrig. He was an American hero in a lot of ways. I'm going to come back to that. But I remember back in the 80s and 90s when I was taking care of patients with ALS. And often would hear that there was so much frustration that this was such a rare disease, and if maybe more people had the disease, if this were more common, that it would get more attention from the pharmaceutical industry, and there'd be more people working on good treatments and a cure. And frustration that maybe ALS didn't get enough attention, and that's a problem with many rare diseases. And one of the things that I like best about working with cell and gene therapies is, if you look at this slide, and at the top of the slide it refers to investigational new drug applications. And you're going to hear me refer to that several times. and I use the abbreviation IND, Investigational New Drug Applications. And these are the research projects that are submitted to the FDA. So when I say IND, I mean a new investigational drug or biologic that has been submitted for clinical trials to the FDA. And in cell and gene therapy, this pie chart, if you look at the area in blue, the great majority of the work in cell and gene therapy is with rare diseases. And that's kind of what I like. I like looking at things for people who too long have been neglected. Now I'm going to talk about several different topics today, starting off with orphan drug designation, which relates to these rare diseases. The FDA has a program for orphan drug designation. The Orphan Drug Act was passed back in 1983. And it provided special incentives for folks who were developing products to treat rare diseases, particularly a disease that affects less than 200,000 people in the United States. That's how we define an orphan drug in the FDA. The benefits of orphan drug designation, developing one of these products for rare disease, market exclusivity. Once the product is approved to go on the market, it limits their competitors for 7 years. There are tax credits. Developing a product to get on the market takes a lot of money. Millions, tens, hundreds of millions of dollars, and lots of different estimates. It varies from one product to the next. It costs a lot of money. And the folks who are doing this now can get some tax credits for that work. Fee exemptions. If a drug company wants to develop a product to go on the market, they have to submit an application to the FDA. Okay, we've got all our data together. Here's all our data. Put our drug on the market. Well, when they submit all their data, there's a little fee the FDA charges. That fee now is over $2 million. But if you're developing a product that has orphan designation, fees are exempt. So these things they count for something. Market exclusivity, tax credits, fee exemptions, they all count for something. And the FDA also has a grants program to help fund some of the research for development of these products. Now this shows what's happened with orphan drug designation since 1983. So in 1983, when the Orphan Drug Act was first passed, and now we have over 3,000 products that have received orphan drug designation. Now those are ones that are in development. They haven't all been approved. We have about 500 products that have been approved under the Orphan Drug Act. Now that's a good start, but we have to realize that there are at least 7,000 rare diseases. And so if we've got products to treat 400 or 500 of those, there's a lot of work still left to be done. Now, there are, as I mentioned, financial incentives for developing a product to treat a rare disease. But the standards of evidence that are required by the FDA are the same for a common disease and for a rare disease. Regardless, we require substantial evidence of effectiveness and some good evidence of safety. We recognize that some things that are done during clinical trials with a common disease are simply not feasible with a rare disease. For example, if you look at trials in diabetes or hypertension, some of those trials have 5,000 patients. You can't be doing that in rare diseases. Some of these rare diseases have a couple of dozen people in the United States with the disease, maybe 100 or 200 worldwide with the disease. We recognize that what's done with common diseases, we can't do that with rare diseases. So the FDA has flexibility in what it requires and the level of evidence. Regardless, the standard is substantial evidence. But what substantial evidence means can be different in different clinical situations. Now I want to say a little bit about natural history studies. Natural history study is a study that follows a group of patients for some period of time and collects data in order to understand the disease. One of the problems with rare diseases is that often no one gets enough experience with it to fully understand the disease. And so we don't really understand the clinical course of the disease well enough to do rational drug development, to know what eligibility criteria there should be for studies, to think about what the outcome measures should be, and how long the studies should last. When someone wants to go into a new disease, particularly a rare disease, we ask the investigators to think early about what is the clinical development program going to look like? What are those trials going to look like? And do we know enough about the disease to design those clinical trials? Now, the FDA can't require a natural history study. We don't have the authority to require it. But if we don't understand the disease well enough, and the clinical course of the disease well enough, then a natural history study can be very valuable for efficient drug development. In 2003, the Human Genome Project was completed. And that project was trying to map all of the genes in human DNA. In 2003, they got to 99% of the genes, and that was their target. There is a revolution in the science of understanding genetics of human disease. And this is happening in labs across the world. So diseases that we used to classify based on what we saw in the patients are now being classified based on the genetics. So the diseases are being redefined. And as they're being redefined, we have to reassess what is the natural history of this redefined disease. All those old natural history studies that we did may not be accurate with the new definitions of these diseases. For example, back in 1990, 1991, I was working in Dallas and just getting started in ALS research. And I worked with a group called the Western ALS Study Group. And we did a natural history study of ALS. This was 30-something years ago. And, or 25. It was a long time ago. and we took about 200 patients with ALS and followed them for a couple of years, just to try to see what the natural history of ALS was. And we looked at patients using 42 different strength and functional measures. So we had patients push and pull with their arms and their legs, trying to look at all the different muscle groups, and we had them climb stairs, and we had them walk for a specified distance, and we did all these different measures to try to understand what the natural history of ALS was. And that study, I think, was valuable in helping to design studies like the one that Dr. Patterson mentioned of BDNF and other studies of growth factors that were done in the mid-90s. So I think those natural history studies helped us. But then folks said, no, we want different outcome measures. These outcome measures, they're not perfect, they take a lot of work. Maybe we can come up with something better. And they came up with something called the ALS Functional Rating Scale, ALS-FRS. And the ALS-FRS has been used for maybe the last 15, maybe 20 years as probably one of the most common outcome measures in ALS. And I'm not here, should I say, to endorse any particular outcome measure. No outcome measure is perfect. Each has advantages and disadvantages. But the ALS-FRS has been used for at least 15 years now. And just a few months ago, there was a publication looking at the natural history of ALS, again, using the ALS-FRS. If you look at ALS using one outcome measure, you get one perspective. If you use a different outcome measure, you get a different perspective. And with each outcome measure comes a different perspective on the disease. So we're starting to do new natural history studies as the outcome measures change. And just a few months ago, there was this publication saying, okay, if we're going to use the ALS-FRS, we need to understand that over 6 months, 1/4 of patients don't really change. And over 6 months, 14% of patients seem to actually get better. Well, wait a minute now. But we've always thought of ALS as this steady, relentlessly progressive disease. Does this mean that that isn't true? I don't think it means that. I think what it means is that if you use an outcome measure, your perspective on the disease depends on the outcome measure. And you have to understand that outcome measure with relevant to the disease if you're going to design clinical trials. Because if you knew this information about using the ALS-FRS and stability over time, then if you were designing a clinical trial to slow down the progression of ALS, you probably need to enroll patients who are going to progress. Enrolling people who are not going to progress is not going to be informative in your clinical trial. So natural history data is essential to efficient design of clinical trials. Now, a couple of years ago, the FDA had a public hearing to hear from patients, patient advocates, academic investigators, the pharmaceutical industry, to hear what the FDA was doing right or wrong. And I think some of the folks here may have actually been at that meeting. And there were a lot of things said. But there are a couple of things that I particularly remember. And one was that patients who have these bad diseases, like ALS, want to have access to investigational agents. And the second was frustration with the fact that we have one product that's been approved for the treatment of ALS. And it extends life by 2 or 3 months. And we've had that product actually now for about 20 years. And 20 years ago, we thought it was a huge step forward. But it's not enough. and frustration that there hasn't been more drug development and more products come along in the last 20 years. So I'm going to talk a little bit about those two topics: access to investigational agents and frustration with the products that have not yet come. So back in the 1980s, there came to the United States an epidemic, the HIV epidemic, patients dying with AIDS, and the gay community really took the lead and said, this is not okay. We need access to new investigational agents. Some of you may have seen a movie that came out, I think in 2014, called The Dallas Buyers Club. It talks about the efforts of patients who were HIV positive to try to get access to investigational agents. And let me say that I don't necessarily agree with the portrayal of the FDA in that movie, but I think it was a fair representation of the frustration and the anger that was in the HIV community and the desperation in trying to get access to drugs. And as a result of their activism came what we think of as compassionate use. At the FDA, we call this expanded access, expanding from just research to actual treatment with investigational drugs. And expanded access is defined by the FDA as use of an investigational drug outside the clinical trial for the purpose of treatment. This is not the purpose of research, this is the purpose of treating patients with a life-threatening disease who do not have good options. Now, we at the FDA like to kind of brag about the fact that if you submit an application for compassionate use of an investigational drug for a patient, we approve 97, 98% of those applications. Not 100%, but the overwhelming majority get approved. And when I talk to my neuromuscular colleagues about this, they always say, but the problem is that the application process is too bureaucratic. It takes too much of the investigator's time. So the FDA, back in last year, a year ago, came out with a new form. It's a one-page form. I will admit it's front and back page. It's a one-page form. It's meant to take about an hour to complete, that can be submitted to the FDA to give approval of compassionate use of investigational drug for a patient. Now, ultimately, the folks who make the product, the manufacturers, also have to agree. Sometimes they don't. Sometimes their product is in short supply and they need all their product to do the research. But the FDA is trying to make this a simpler, easier process for patients and their doctors. Now I want to come back to Lou Gehrig, because Lou Gehrig, of course, we think of as an American hero, and part of what made him a sports hero was that he was a home run hitter. And so I like what Dr. Patterson said a minute ago, that they're going into drug development with the idea of trying to hit a home run. Because heroes try to hit home runs. One of the things that I like most about working in cell and gene therapy is that there are doctors out there, and this paper comes from Carl June's group, and Carl June is, I think, at the University of Pennsylvania, right up the road, and is looking at a genetically modified cell for the treatment of leukemia. And just this little snippet from the abstract says, with complete remission. I hear people talking all the time about they're trying for an incremental change. And I hear that and I just cringe. And I think, are you telling me you think your drug really doesn't do much? That's not how we're going to fix these diseases. People need to try the home run. And there's some folks, like Dr. June's group, like Dr. Patterson, who are talking about home runs. This is a paper from Jerry Mendel and his group, looking at a rare genetically defined muscular dystrophy. And in his abstract, he talks about the potential for reversal of disease was displayed. So often I hear people talk, particularly in neurodegenerative diseases like ALS and Alzheimer's disease, oh, we're going to slow down the progression. Sometimes they'll say, we're going to stop the progression. That would be wonderful. That is a home run. But every now and then, there are folks who will say, no, we're going to go one step further. We're going to reverse the damage that was done. And I love this. These are my heroes. The people who are saying, no, these things can be done. But if we're going to do it, it requires collaboration. And certainly, Dr. Patterson has mentioned in the multidisciplinary clinic, taking care of the patients on a day-to-day basis is a team effort. Well, doing the research is also a team effort. And this doesn't list all the folks who are involved. But the people who are involved, the scientists, the clinicians, the folks who look at outcome measures, the folks who take care of these patients, the people who fund the research, the industry, the NIH, people have to talk to each other. And they have to talk to each other from the very beginning. As soon as somebody gets an idea, they need to reach out to the other people that are involved here if we're going to have efficient drug development. Can't wait until the last minute. Can't wait until after you've done your animal studies and you're ready to go into the clinic to start talking to the advocacy groups, to start talking to the pharmaceutical industry. Early collaboration is essential. Now, I've talked about gene therapy and rare diseases. And you think, well, what does this have to do with ALS? And speaking of what does this have to do with ALS, these two folks, this is Joseph Goldstein and Michael Bryan, two gentlemen who were interested in studying cholesterol. And they found some individuals who had a rare genetic form of high cholesterol. And they studied these folks, and they eventually, in 1985, got the Nobel Prize for their work. But what their work led to, again, based on studying a few individuals with very rare disease, their work ultimately led to the statins, which are now administered to millions, probably tens of millions of people in this country. There aren't tens of millions of people with that rare high cholesterol, but what you can learn from studying a rare genetic disease can lead to innovations that affect millions of people. And we all know that there are rare genetic forms of ALS. And there are folks who are studying those diseases. And what we hope is that by understanding the genetics of those diseases and finding treatments for those diseases, it will extrapolate to treatments for other patients with ALS, other patients with perhaps even other neurodegenerative disorders. So this slide shows what's been happening with gene therapy. And I'll come back again to the very top of the slides, gene therapy INDs. INDs are investigational new drug applications. So this looks at the number of new INDs for gene therapies that have come to the FDA since 1989. Was when the first one came in. And if you look from 1989, on the left of the screen, you'll see that this sort of rapid increase up to 1999, where there's this kind of peak standing out here. This rapid increase. And then, unfortunate things that happen. As with any new field, there are going to be setbacks. There were some setbacks in gene therapy. Some patients were injured, and the field dampened quickly, so that by 2002, the level of gene therapy INDs was less than half of what it had been in 1999. But the science has advanced rapidly with the Human Genome Project, with development of new and better ways of delivering gene therapies. You'll see that from 2002, by 2012, it took 10 years, but we got back up to the same number of INDs as we had in 1999, that previous peak, and we've kept going. So if you look at 2015, last year, we had twice as many new gene therapy INDs as we had in 2010, and almost 4 times as many as we had back in 2002. So gene therapy is moving quickly. And in the science, this is looking at clinical drug trials. The science is moving even faster. And we all believe that as this science moves and the clinical research will continue to grow, that we will find treatments for these rare genetic diseases. And what we learn from these rare genetic diseases will hopefully lead to treatments for other diseases and more common diseases, and particularly for ALS. Now, I need to acknowledge the folks in my division who do all the work, and particularly for neurologic disorders, Lei Zhu is an MD, PhD who handles many of our neurologic applications. And I'm going to stop there. If you have any questions, there's my email address, and I'll be around for the rest of the next hour or two to answer questions. But I do appreciate the opportunity to talk to you. I think there's a lot of excitement that's going on in gene therapy. There's a lot of excitement in research, and I think that it's good science that ultimately will bring the answers. Thank you. Oh, you want me to? Sure, I'll take questions if you have any questions.
So then I'll just give a comment, because I think, you know, I made a comment because I wasn't doing an ALS talk, but you heard Dr. Bryan mention gene therapy. This is directly related to ALS. There have been now, we used to say, oh, only 10% of ALS is familial. We've now identified more than 30 underlying genetic defects that can lead to the development of ALS. We found that some of them, in particular, are quite common and can even occur in people who had no family history, defining that there are genes that make you more susceptible to developing ALS and therefore would be targets for treatment of ALS. And what's even, oh God, I'm hiding myself again. What's even more provocative, there is an undercurrent within the research community that in fact we are who our genes are. That in every case of ALS there is a genetic component, and more that is becoming extremely prevalent as an opinion. And certainly I do feel that your genetic endowment plays a role. And what does that mean vis-a-vis therapy and targeting? It means my dream someday is that we will define a person's genome when they develop ALS and be able to pick out those genetic abnormalities that might be targetable for therapies. And we have the tools, which is what Dr. Bryan was intimating. We have the tools now to really go after gene therapy, to be able to deliver it in many ways at many levels. Not only deliver the gene that's abnormal to a person, but actually modify how that DNA is translated, made into RNA and made into proteins. That's what the oligo community is doing. And in fact, in ALS, there are already trials of antisense in SOD1 mutation patients. These are not far-fetched. It's not tomorrow, it is today, and we're making real progress. So I want that hope, you guys to understand how hopeful we all are that we will begin to really crack these codes. Yeah, Frank.
Hey, do all ALS patients show elevated glutamate levels, or is it just the ones that pathway?
Well, yeah, not all do, but that pathway is active. If you take a group of ALS patients, and this has been done long ago, we haven't really done it on everyone, and you look at the levels of glutamate, they're slightly elevated. We looked in the animals. In fact, Bill did some of the microdialysis work in our animal models and showed that there was some poor reuptake of glutamate. So, that's one piece of the picture. It may not be, it may be downstream. There may be a cascade, a domino effect. Something starts the disease in any individual, and then, like anything, we only have, our body, our brain has only certain ways it can respond to an insult. And so once the insult begins, and perhaps that's with a genetic mutation and some aberrant process in our motor neurons, then the dominoes fall and that whole cascade of cell death is initiated. And one of the reasons that riluzole, which is the one drug available, may not be the home run we would hope for. Maybe that it targets a downstream process that the horse is already out of the barn. And I don't know if you want to comment on anything I just said. Or...
No, I didn't go into the specifics, but what Dr. Patterson said is actually 100% accurate. The science is really moving forward very rapidly. We're developing new ways to deliver gene therapy, and we think that as we understand the genetics of ALS better, we will have more and more targets. Now, which ones will be the right targets? I don't know. I don't think any of us know. But we have to explore all these different avenues in order to find those treatments.
And the FDA is our friend, right?
Yes.
No, and I think it's really important for you to understand that the collaboration between the investigator and the FDA's job is to protect people and to promote drug mobilization into the clinic and to people, but they want that to be done safely. They are not our enemy, they are our partner in this, and that's important for everybody to recognize. I thought people, do you want to take a little stretch and then we'll start our awards? People have to, anybody need take 5 minutes to stand at least, and we'll start in 5 minutes.